going a bit deeper into transcription and retrotranscription, if we remember the course of the last week, transcription is then the process to read DNA by RNA polymerase. We have here the DNA. We have this pink protein is the RNA polymerase, and DNA is unzipped, like opened by a protein called helicase. This helicase is important for our next diapo. So helicase opens or unzips this DNA, and in order for DNA can be read and RNA can be constructed from this DNA template. We have the non-template strand and we have the template strand, which is the one that are being transcribed to or copied. And the direction of the transcription, as you can see, RNA is constructed from 3' to 5', and we have several other proteins helping the DNA structure to rewinding and unwinding, and that kind of twisting. When you open the DNA, when you unzip it, you create a twist force. So proteins need to be added there, so this DNA twist do not have problems. And as you know, adenine is when we're transcribing, adenine is paired with uracil, cytosine is paired with guanine, and this is pretty much the problem. So we're going to have Howard Wolowitz to give us a couple of examples of how you can use this knowledge to talk to a girl or a boy in a bar. So you can arrive and say, I wish I was adenine so I could get paired with you, haha. And you can also say, if I was an enzyme, I would like to be helicase so I could unzip your genes. Well, okay, enough of jokes. Let's continue. We have another process called RNA processing. So when transcription has created this RNA molecule, it reaches different kinds of processing or RNA editing. For example, in here, what we've talked about is we have this portion of the RNA called exon 1 and exon 2 and exon 3 and so on, and we have these parts here called intron, that are intron 1, intron 2 and intron 3 and so on. So this RNA is called the primary transcript because it's the exact copy of the DNA. And when we have this process called splicing, that is when exons or introns are cutted out from the primary transcript and exons are pasted one behind another. So this is the splicing step. We also have messenger RNA 5' capping. This capping is it's a portion added to the 5' extreme of the RNA. This has many functions. The first of all is the regulation of nuclear export. These pieces here help to export the RNA. We also have this 5' capping can also help in the prevention of the degradation of the RNA, because we have these mean proteins that are called exonucleases that cut into pieces these RNAs. So when they found that they have this cap in the 5' extreme, they don't cut it. So this could help to the RNA to reach the ribosome. 5' cap also promotes translation. And I think that's it. Okay. After this 5' capping, we have another step called polyadenylation. That what happens is that in the 3' extreme, a poly A tail is added. We have a lot of adenines here that are added in this extreme. It's very important also for the nuclear export, for translation, and for the stability of messenger RNAs. This tail is shortened over the time. This will be because a messenger RNA, when it reaches the ribosome, it can be translated many times. So many proteins can be done from this message. However, this poly A tail is being shortened in the time, and when many copies are made, and when it is short enough, mRNA is enzymatically degraded, which means that it's cut into little pieces, and the nucleotides are or could be reused. We have several kinds of RNAs. We have, for example, the messenger RNA that carry protein sequence information to ribosomes. This is the main RNA that we've been talking about. We have transfer RNA. tRNAs are the ones that can translate to a nucleotide information to protein information. We're going to talk about tRNAs a bit further. We have ribosomal RNAs that are catalytic parts of ribosomes. They are like parts of the big machine called ribosome. We have microRNAs and small interfering RNAs that could be downregulators of gene expression, because as you can see here in this draw I made, miRNAs or siRNAs can block regions of messenger RNAs by combining to them, by pairing with the sequence we have in an RNA. So it's blocking. It can block the expression of this RNA, the translation. We can also have small nuclear RNAs that are involved in splicing, transcription, and telomere maintenance. I'm not going too deep into telomeres. We have small nuclear RNAs that perform modification of RNA nucleotides. So, and these are just the most common RNA types. We have a lot more different RNAs that we don't even know all of them and what they function.